Our case study presentation is on DNP and it is done by Kimberly, Isabella, and Jacob. Connor is on the wrestling team and wants to lose a few pounds to make into a lower weight class. He asked his friends at dinner if they had any suggestions on a quick way to lose weight. They told him about an unregulated drug called 281 in the 1930s. His friend Becky warned him to be cautious of the dosage. Connor looked into it and found some information about this diet pill now called DNP. The information stated that it helped a lot of people lose weight, but other side effects include fever, cataracts, and rashes. Some reports included death. DNP is said to be a mitochondrial uncoupler. This is a molecule for DNP. If you have taken organic chemistry, you've probably learned how to name molecules. This molecule is 2,4-dinitrophenol, dinitro for two nitro groups, and 2 and 4 for its position in relation to the alcohol group. DNP is also sold under several different names, all promising the same miracle weight loss effects. It is also used in pesticides, wood preservatives, dyes, and explosives, but as a miracle weight loss drug, it has side effects of fever, cataracts, which is clouding of the lens causing blurry vision, and rashes. It is also a mitochondrial uncoupler that takes away the energy from the breakdown of fats and sugars, which we will discuss later in this video. An important thing to note is that DNP is not approved for clinical use, which we will also learn about why that is in a little bit. To start off, we need to understand a little bit about the mitochondria. As we all know, the mitochondria is the powerhouse of the cell. So what does that mean? If we think back to cell biology, we know that the mitochondria is in charge of making ATP, which is energy that powers all of our bodily functions, hence the powerhouse of the cell. Specifically, the electron transport chain is what takes pyruvate made from glucose in the glycosidic cycle and convert that into ATP for the body to use. To better understand this process, we must think about the structure of the mitochondria and where the electron transport chain takes place. There are two membranes in the mitochondria, an inner and outer membrane. As you can see in the diagram, the area between these two membranes in blue is the intermembrane space. Within the enclosed space of the inner membrane in red is the matrix. The electron transport chain and site of oxidative phosphorylation occurs in the inner membrane of the mitochondria, where a proton gradient is created by electron carriers and complexes. The diagram shows the protons being pumped from the matrix into the intermembrane space. This proton gradient is an important part in ATP synthesis and where DNP plays a role as a dangerous weight loss drug. Now that you know some of the basics of where and how the electrons move, let's dive into the details and see how DNP plays a role.
Now that we reviewed some of the basics, let's get down to the nitty-gritty. Each of the embedded structures are called complexes, so complex 1, 2, 3, and 4, and then at the end over here we have our ATP synthase. Each of the complexes have their own names, but for the sake of simplicity, we're just going to be referring to them as complex 1, 2, 3, and 4. The purpose of these complexes is to pump hydrogen ions into the intermembrane space by using hydrogens attached to NADH, or in the case of complex 2, FADH. This creates a hydrogen ion gradient where there's a high concentration of hydrogen ions in the intermembrane space and a low concentration of hydrogen ions in the matrix. Hydrogen ions will want to go down their concentration gradient, but the only way to do that is by going through ATP synthase. Hydrogen ions power ATP synthase and essentially turns the wheel to add a phosphate group to ADP, making ATP, while also releasing hydrogen ions, therefore allowing them to follow their concentration gradient. During the process, in order to remove hydrogen ions from NADH and FADH2, electrons from that bond must be transported through the electron transport chain. This is done by two proteins which are associated with the electron transport chain, which are cytochrome C and coenzyme Q. So these two electrons from the bond are transported through the electron transport chain to first coenzyme Q, and then to cytochrome C, and eventually to our byproduct, which is water. And with each stage, they're decreasing in energy state. So now that we've had a little bit more of a review of the electron transport chain and how it works, we're going to see how DNP affects it. So DNP is a mitochondrial uncoupler, and that will explain what this means in a second. So DNP embeds itself into the membrane of the mitochondria and serves as a mitochondrial uncoupler. All uncoupler means, in this case, is a way to bypass ATP synthase. So as we see here, here's DNP that's embedded itself into the membrane. Here's ATP synthase, and we see that those protons are using the DNP complex to bypass ATP synthase. As we went over, complexes of the electron transport chain pumps hydrogen ions into the membrane space, therefore creating a hydrogen gradient. Traditionally, these protons will want to go down their concentration gradient and will go to or go through ATP synthase, therefore creating ATP. However, the DNP embedded into the matrix, protons are able to essentially take the highway out of the intermembrane space. This reduces the overall concentration of hydrogen ions in the intermembrane space, therefore limiting the amount of ATP that can be produced. So we're having a decrease in concentration in the intermembrane space because they're able to take DNP out. And it's essentially the path of least resistance with the DNP versus the ATP synthase. So they're able to quickly get away from each other because they're all hydrogen ions through DNP, therefore reducing the amount of hydrogen ion concentration we have in the intermembrane space. And when that decreases, ATP synthase will not be able to make as much ATP. So we're going to get into another way to look at it. Essentially, we're going to be using an analogy. We're going to call this a highway analogy or the toll booth analogy. So in this case, our ATP synthase is the toll booth. The toll booth separates the two areas. So in the cell, that would be the matrix and the intermembrane space. And the DNP is the highway that bypasses the toll booth. So on a normal road with a toll booth, usually everybody will be going along and they have to pass a toll booth. They have to pay the toll booth, just like the hydrogen ions have to go through the ATP synthase, and they have to help produce ATP for them to get through to go down their concentration gradient. Same things happen here. Cars have to go through the toll booth, they have to pay, and then they're able to go through and go wherever they want to. But this can get quite backed up and it takes some time. That's what essentially powers the ATP synthase. We have all these hydrogen ions in the intermembrane space wanting to go through ATP synthase to get away from each other, to go down their concentration gradients. Same things with cars. They want to get out of traffic. They're trying to get through the toll booth. They pay the toll booth. They're able to go through. Hydrogen ions help make ATP, able to get through. But if people are smart, they'll take a highway that bypasses the toll booth, which is essentially what DNP provides. It's a highway to bypass ATP synthase, allowing them to go down their concentration gradient, which is a whole lot quicker, and therefore we won't produce as much ATP. So if we're not producing as much ATP because DNP is lowering our intermembrane concentration of hydrogen ions, what do we do? Well, the answer to that is going to be glycolysis. With the electron transport chain being compromised, the only way to supplement the loss of ATP that was being produced is through glycolysis. Instead of producing 32 ATP like we were with the electron transport chain, now we're only producing 2 ATP with glycolysis, meaning we're going to have to break down a lot of glucose just to break even here. Since we're not eating all the time, and we don't have a steady stream of glucose, the only way to increase our glucose concentration is through gluconeogenesis, which y'all should remember what that is from human phys, the breaking down of fat deposits to form glucose, which will then go through glycolysis, which again gives us 2 ATP. Because of the breaking down of fats for energy, overall body weight goes down, therefore doing exactly what Connor wants it to do, which is getting rid of the fat equals weight loss is exactly what he means, right? But now let's take a look at some of the data regarding DNP and if it's really safe to use and what about some of the implications because of that. Now that we got a good look at how DNP works as a mitochondrial coupler and the mechanism for that, now we're going to get a little bit of a broader look and look at DNP in data form. So this graph is showing us the effect of DNP on muscle cells, which we all know are heavy users of ATP. Over here in our control group, we have our ATP, and on our y-axis right here is the percent of control. So it's a control of ATP and our control of glucose oxidation. And so we see that they're just about in relative levels. However, when we look at the group that's been treated with DNP, we see our ATP percent control go down quite a bit, a decent amount, and our glucose oxidation skyrocket. So essentially what's happening here is that we know DNP is a mitochondrial coupler. It's allowing the uh, hydrogen ions in the inner membrane space to go down through the DNP complex and bypass ATP synthase, therefore dropping the amount of ATP we're producing through the electron transport chain by a lot, which we see here by that little divot or dip, I should say. But notice how it doesn't go all the way down, and that's because we're using glycolysis, we're breaking down the fats, we're forming glucose to then break down into ATP at a rapid rate, and that is what's shown here by the glucose oxidation. Our glucose oxidation skyrockets to try to make up for that all the ATP that we're losing or not producing from the electron transport chain. And so it's trying to stabilize our ATP. That's why our percent of control of ATP goes down a little bit, and our percent of control for the glucose oxidation skyrockets because that process is being heavily upregulated to make up for the loss of ATP from the electron transport chain. Now that we discussed both the mechanism and some of the data, we're going to be looking at some of the implications of DNP and looking at it from a broader aspect and its other effects on the body.
Now that we have looked at the mechanisms and data of the specific case study, we are going to explore various implications that DNP can have on the human body. For example, fevers are one of the effects of DNP. Instead of producing ATP with the proton gradient as it moves down the concentration gradient, DNP loses energy in the form of heat. Priority is placed on the production of heat instead of the production of ATP. In addition to fevers, there are several other effects DNP can have on the human body. When looking at the New Jersey Department of Health and Senior Services Hazardous Substances Fact Sheet, the hazard summary describes the effects of DNP in detail. In addition to causing a fever, DNP can cause damage to the kidneys and liver as well as damage red blood cells to the point of causing anemia. When a patient experiences repeated exposure to DNP, damage to the eyes can occur, which can lead to the formation of cataracts. Rashes and dry skin can also be a possible side effect. There are numerous other effects of DNP exposure, such as damage to the nervous system, which are listed in the image to the right of the slide. Evidence such as the summary sheet indicate how harmful DNP effects can ravage the human body and emphasizes the severity of cases such as Connors. As several of the harmful side effects were just previously mentioned, it is important to understand how these symptoms can build up to a point where the effects are irreversible, causing death. Patients experience hypothermia, which is a significant decrease in body temperature, along with metamyoglobinemia, which is a blood disorder where there is too much metamyoglobin in the blood, which is a type of hemoglobin. This disrupts the process of oxygen going to various parts of the body. DNP effects can become even more severe, such as when cardiovascular collapse happens. Cardiovascular collapse occurs when there's a severe form of hypotension or low blood pressure. This causes loss of consciousness, heart arrhythmias, which are irregular heartbeats, along with other serious heart issues. This condition ultimately results in death. So once cardiovascular collapse occurs, this is the last thing that would occur before the patient would die, and this is to the point where DNP effects have become so severe that it is irreversible. In order to bring some further context to how DNP has caused these significant issues within human health, we have compiled a list of a few facts that will help contextualize how DNP impacted the field of medicine and the study of biochemistry. DNP originates from its use in French manufacturing during World War I. It was also prescribed to soldiers for warmth in World War II. DNP could be found in substances such as wood dye, photographic developers, and herbicides. The first death that was reported as being caused by DNP was in 1918, and prescriptions for DNP stopped in 1938, as it was deemed not safe for clinical use. All of these facts about DNP provide context as to why DNP could be dangerous to Connor, and how biochemical mechanisms within the human body interact with DNP to form a potentially deadly combination. In summary, DNP is a weight loss drug that is not approved for clinical use due to its many harmful side effects. Oxidative phosphorylation in mitochondria as the mechanisms are the biochemical factors that play a significant role into how DNP negatively interacts with the human body.